And full screen, I will also put it there. So I hope that now you see the presentation.
Yes.
Perfect, perfect. As you said, I just have to down this. Okay, perfect. So the name of our presentation and our work is use of immunofluorescence quantum dots microscopy analysis for the detection of food allergenic components in meat products. Dr. Tremblova is the main author of this research, and we are all coming from the University of Veterinary and Pharmaceutical Sciences here in Brno in the Czech Republic. And actually this work consisted of work was done on two departments, the Department of Plant Origin, Foodstuffs, Hygiene and Technology, and Department of Biology and Wildlife Diseases. Actually this work was also the part of one doctoral thesis. I will start immediately about food allergens, and at the beginning I will just stress out that food allergy represents a very important issue worldwide, since the population sensitive to food allergy is increasing. The awareness of this problem is increasing among consumers, but also among producers and among control agencies that need more rapid um, methods, and they also need reliable methods. Also, we are facing that different laboratories have different equipments, and some methods are more suitable for one laboratory, other methods are more suitable for some other laboratory. Food allergens also is the fact that more and more children have problem with food allergy, and also people in adolescence or old age can you very often develop a certain food allergy, and they have problems. What is the problem with food allergy is that the quality of life of those people are also affected, and they have to rely a lot on food labeling. Labeling that here in the Europe is focus on mainly regulation 1169 2011, and also Codex Alimentarius. What is also important is that, as I said, is that labeling, and this labeling is under influence of true claiming guaranteed by the manufacturers, and according to regulation, it is due to each producer to truthfully inform consumers about the consumption of their food product. Failure to provide all correct information about composition and about components in food actually is declared as food adulteration. So the problem is that food adulteration and not declaring allergies to allergens on the labeling can cause a serious problem to people with food allergy. Why it is happening? It is usually happening before certain producers are trying to actually replace certain components with cheaper ones, and it is very bad when it is done by ingredients that can be allergens, and for some people can cause serious health problems. When we are speaking about the detection of allergenic components in food, there have been developed numerous methods, and they are still evolving. Approaches to method development are constantly evolving with respect to the metrics of food. It is also important to say that the metrics of food is very important, and what kind of food we are going to detect allergens in them. As you know, even with other food matrices and other methods when we are doing certain analysis always is the problem that specificity and different foods are reacting differently to certain methods. There is no single method that suits all purposes. So when we are speaking of food, usually it is like that. And also these methods can be done qualitatively and quantitatively. Our method was based mainly to qualitatively find the presence of allergens present in our model samples. But our method also can, and it is going to be our further research, can be done in the way that we will try to perform quantitatively our method. Following methods are preferred for detection of food allergies. Immunoassays, ELISA and PCR-based methods are very often used, and that's why in our research we did actually as it served as control samples actually that we compare ELISA methods with our immunohistofluorescence quantum dots method. Other methods are also used in the detection of allergenic ingredients, chromatographic methods in conjunction with usually mass spectrometry, and also others. The aim of the research was the development of suitable immunohistofluorescence quantum dots methodology using cadmium selenide and zinc sulfide quantum dots as a new type of fluorophore in immunohistofluorescence labeling of allergens in meat food matrices. Gaining new knowledge in the possibility of using commercially available conjugates of quantum dots with bond secondary antibody as a marker suitable for microscopic detection of allergen, wheat protein gliadin and milk protein casein, and also verification of immunohistofluorescent quantum dots methods on products purchased from a market network containing allergenic components of wheat and milk. We also did that verification with certain amounts of samples with samples from network, from markets, and actually we did a little survey of the market, how truthfully meat products are declared that they have allergenic components in them or not. Comparison of the development method with ELISA method, of course that we had to compare our method with present ELISA method, which we can say that are reliable for these kinds of detection. Analyze materials, we did model meat products and around 28 samples we did it, and commercial samples, it is small size, 50 samples, but we wanted to check it, and we used heat-treated meat products. Actually, we also produced model samples heat-treated meat products here in the Czech Republic. Heat-treated meat products, it means that the product was heated at 70 degrees of Celsius at least during 10 minutes, and 70 degrees of Celsius has to be in all part of that product during at least 10 minutes. And actually that product is ham. Ham here in the Czech Republic is very popular. Model meat samples were prepared from minced chilled pork and beef in a ratio 1:1, 50% of pork and 50% of beef meat. To improve binding, we added 1% common salt in the mixture, and we made 9 concentrations of allergens from 0 to 10% with calcium caseinate and with wheat protein. Model samples, as I said, were hams cooked in a thermostatic pot with a temperature setting at 90 degrees of Celsius. When we reached 70 degrees of Celsius in the core of the product, they were cooked for 10 minutes while maintaining this temperature. The detection of milk protein and wheat protein we also did in the samples from the market. Meat products were purchased in the market network, and samples did not contain any declared allergenic ingredients. In the list of ingredients were considered as negative commercial samples, and positive samples were those declared that they contain allergenic components. What we also did, as I said, we compared our method with ELISA methods, and two types of commercial kits were used: Ridascreen Gliadin and Ridascreen Fast Milk. The procedure of the methods were performed according to the planned detective target group of allergenic components. Ridascreen Gliadin ELISA kit is an immunoassay with the sandwich enzyme for quantitative analysis for chrolamines. From wheat to also rye and barley it is used. And it is used to control the contamination of diet products in patients usually with celiac disease. Ridascreen Fast Milk is also sandwich enzyme immunoassay for the quantitative determination of milk proteins in foodstuffs. Antibodies specifically detect caseins and beta-lactoglobulin from cow's milk, but also this method is used for detecting allergenic components in sheep, goat, and buffalo milk. Immunohistofluorescence quantum dot methods. It was done in the way that samplings from our samples were taken from 6 sampling points, and processing was done using the technique of paraffin blocks. During the development of the new immunohistofluorescence quantum dot methods, 3 types of quantum dots were tested. And once the samples were impregnated with a fixative solution for at least 24 hours, the next step, they were dehydrated with an ascending alcohol series, ethanol we used, and subsequently saturated in a heat paraplast. The temperature was 56 to 58 degrees Celsius. The drained and saturated samples were placed in potting boxes and then embedded on a watering machine. And liquid paraffin was at a temperature 58 degrees Celsius. 4 paraffin blocks were created for each samples. Histological sections were deparaffinized prior to immunohistofluorescence quantum dots and immunohistochemical examination. Evaporation was performed in xylene and alcohol baths. The principle of histofluorescence quantum dot methods can be seen actually on this presentation, on this slide. And primary antibodies labeled with commercially purchased conjugates of secondary antibody against rabbit FC fragments with bound CD, SC, and ZNS quantum dots with emissions, actually peak fluorescence of these quantum dots were 525, 585, and 655, and that contrast of these antibodies, contrast with antibodies and background. Actually, we qualitatively tested with fluorescent microscopy, which is actually shown here. Then the selection immunohistofluorescence quantum dot methods we perform in the way that we choose for the selection of fixing agent. We use 10% of formaldehyde. That is the for us, it was the best solution. The primary antibody dilution was in a ratio of 1 to 300 and 1 to 500. Mounting medium for immunohistofluorescence quantum dot methods, we used solacryl for mounting medium, and for the meat products, it was actually also we tested the best solution for us. Commercially purchased conjugates with peak fluorescences with 525, 585, and 655 nanometers. We tested which one has the highest contrast with background, so which one would be the best to perform and to find that allergenic components. On this figure, you can see that comparison with quantum dot 655, quantum dots 585 and 525. These less darker or light are actually test negative samples. These ones are positive samples, and we can see that quantum dots 585, they showed the biggest and the highest contrast in comparison with our control samples without allergenic components. So the comparison of contrast between conjugates with different types of quantum dots are shown here, and that's why we actually choose quantum dots 585 for further detection. Also, this picture is showing even better that the highest contrast we performed with quantum dots 585 conjugate, because here on this picture 1 and section 2, the highest contrast can be seen was performed with conjugate 585. In the comparison, in the comparison immunohistochemical method and immunohistofluorescent quantum dots 585 methods, we can see also contrast with background with our quantum dots of wheat protein, and also here with caseinate is shown here that this contrast is relatively high. And now we can see finally the comparison of qualitative results in the identification of allergenic components of milk and wheat in model meat products. As I said, we used immunofluorescence quantum dots 585, and ELISA method was performed for as actually control method. The addition of allergen was from 0 to 10%, and when we check this table, we can see that actually only in this lowest concentration of allergen, so it is about actually the detection and detection limit of our method, that there we had certain we can say problems. But in other concentrations, all our results are in comparison with ELISA actually the same. The same situation is here is caseinate and here is gliadin, and the same situation is with gliadin. With other higher concentrations, 0.001 to 10%, we had the same. We had actually the same finding as with ELISA, with ELISA content, ELISA method. I will stress out again that actually this method it is just a qualitative method. We just found that that allergenic component was present there. It is not still quantitative method. Analysis of meat products purchased in the market network. The correlation also between the microscopic immunohistofluorescent quantum dots 585 method and the ELISA method was determined to be 85% in the analysis of allergens in meat products, which is also good finding, and also no statistical difference was observed. So same as with model samples, with our samples purchased from the market, we found high correlation, 85%, and we found we confirmed that our analysis as qualitative analysis can be used reliably for the detection of these allergenic components. Quantitative analysis, quantitative analysis of ELISA method. We just decided to take maximum value considered 0 when gluten was under 20 milligrams per kilogram, milk protein 2.5 milligrams per kilogram, maximum value considered as a trace amount, gluten 50 milligrams per kilogram, and milk protein 25 milligrams per kilogram. Here is also written the sentence about our samples from the network that we found 25% of tested samples of meat products. 50 samples we tested, and 12 out of them did not comply with the manufacturer's declaration. So with this finding, we are also stressing out how is a big issue that not declaring all components of food ingredients even here in the Europe, and these not declared allergenic components can make big problem for people with allergies. Also, what we did the quantum stability of quantum dots conjugate over time period, and with grayish line is showing, and also it is a quantum dot 585. Grayish line is showing the stability at room temperature, but on the light we see that stability is not so high. Orange line is showing quantum dots 585 stability on room temperature, but in the dark, so that stability is a little bit better. And also bluish line is showing actually the best stability of quantum dot 585. So we can recommend storage for quantum dots 585 to be in refrigerator at temperatures from 2 to 8 degrees of Celsius. So it is actually the best. It is show the best stability on these temperatures. Conclusions according to our results. Immunofluorescence quantum dots method applied to the analysis of 2 types of allergens was compared with routinely used ELISA methods. Quantum dots can be even with our research considered as suitable tool for labeling allergens in food metrics, but for now just quantitatively. The emission intensity of quantum dots, which gave a high contrast to background in the application of 585 quantum dots conjugates, was also evaluated, and we found that quantum dot 585 gave the best contrast. This high contrast of quantum dots 585 with allergenic components can be used mainly for quantification of the results. So we are planning to apply image analysis for future research studies, for example. This immunofluorescence quantum dots microscopic method can be considered a reliable tool for the rapid detection of gliadin and casein in commercial food products, actually. PCR and ELISA methods, which are broadly used for the detection of allergenic components, but we can compare our method with PCR and ELISA methods by looking at 4 major elements. So price, how that method is time consuming, technical skills, and laboratory equipment. So the biggest disadvantage of our method is actually the price. So it is more expensive than ELISA and PCR, but that thing can change over time, and also it is more time consuming. On the other hand, what can be maybe declared as advantage of immunofluorescence quantum dot methods is that for certain laboratories already possessing fluorescence microscopy, this method can be better for them because they already have skills, technical skills to perform this method. What is another advantage of immunohistofluorescence quantum dots method is the possibility of storing already evaluated samples. So we are not destroying our sample with immunohistofluorescence quantum dot methods, and we can repeat the process, repeat the detection in the next few weeks without problem. This experiment can be followed up by further studies to detect other allergens and also to quantitatively found what is the exact amount of allergenic components by immunofluorescence method, and also by our method, as I said, those numbers of analyze samples from the network is certainly not enough, but out of 50 samples that you took from the market, that already 12 are declared not properly, actually is an alarm that allergenic components are very often present in our daily food. So for people suffering certain allergies can be considered a big issue, very for them it is a problem. And also this method is showing that quantum dots can be used for the detection of allergenic components, as I said, quantitatively for now, but certainly that our department will continue with quantum dots, immunofluorescence quantum dot methods, and we are already did some detection with other allergenic components, but also we are working on qualitatively methods. So I would thank you for your attention, and it would be all from me.